Good evening, everyone. Today's topic is about intracellular accumulations, pathologic calcification, and cellular aging. Accumulations are certain things which get accumulated within the cell. And one of the manifestations of metabolic derangements in cells is the intracellular accumulation of abnormal amounts of various substances that may be harmless or it can be associated with varying degrees of injuries. The substance may be located in the cytoplasm, especially within the organelles, typically the lysosomes, or in the nucleus, and it may be synthesized by the affected cells or may be produced elsewhere. There are four main pathways by which the intracellular accumulations will accumulate within the cell. See, where and all can you have accumulation? If we use common sense, we will know that certain things will accumulate once there is abnormal metabolism. That means it has to be degraded. There is a fault in the degradation. So inadequate removal of a normal substance secondary to the defect in the mechanism of packaging and transport. Okay. Second is accumulation of an abnormal endogenous substance as a result of either genetic or acquired defects in the folding, packaging, transport, or secretion, as with certain mutated forms of alpha-1 antitrypsin. So this is where it will lead to accumulation of alpha-1 antitrypsin deficiency, in that there's accumulation of these proteins. Next, where all it can accumulate, if there is a failure to degrade a metabolite because of inherited enzyme deficiencies. The classical examples are lysosomal storage disease and the glycogen storage diseases. And fourth one is where there is deposition and accumulation of an abnormal exogenous substance when the cell has neither the enzymatic machinery to degrade the substance nor the ability to transport it to other sites. Accumulation of carbon or silica particles is an example of this type of accumulation, because it is basically an exogenous pigment and our body doesn't have any machinery to degrade that. So let's see some of the accumulations which are prominent and which may cause pathological significance. Some of them do cause disease. Some of them are seen on our histopathology section, so that we will identify them and we will call them with certain names, so that the clinicians are familiar with that and they know what is the significance of finding such a histopathological finding in the sections. First is lipid. One way in which the lipid will accumulate is called steatosis, that is fatty change. This term is used to describe abnormal accumulation of triglycerides within the parenchymal cells. Fatty change is often seen in the liver because it is the major organ involved in fat metabolism. But fatty change can also occur in the heart, muscle, and the kidney. The causes of steatosis include toxins, protein malnutrition, diabetes, obesity, anoxia. And which is the most other commonest cause of fatty liver is alcohol. So alcoholic liver disease starts with fatty liver. That's the first change. Then it goes to alcoholic hepatitis. Then it goes to cirrhosis. So fatty liver is the first step in alcoholic hepatitis. Also, it is associated with diabetes and obesity. Obese females will always have fatty liver. That's the microscopic picture. Fat gets dissolved in our sectioning of histopathology processing specimens. So what happens is that wherever the fat was there will appear like an empty space. This is the liver. Okay, these are the hepatocytes, and this here is the fat. Okay, this is a fat cell. This is what we call as a fatty liver. The other form in which the lipid will accumulate is with cholesterol and cholesterol esters. What are the conditions in which we are going to see this? One is atherosclerosis, second is xanthoma, then cholesterolosis, and then Niemann-Pick disease type C. What happens in atherosclerosis? You see that there's going to be an atheroma. Now, this atheroma is basically an atherosclerotic plaque which consists of smooth muscles and macrophages within the intimal layer of the aorta and the large arteries, and they are filled with lipid vacuoles. And basically, what are these lipid vacuoles made up of? They are made up of cholesterol and cholesterol esters. So that is what is going to make a atheroma. And Once we see the histopathological section of such an atheroma, you will see that there's a foamy appearance of these cells. The macrophages, which are laden with cholesterol and cholesterol esters, will appear foamy. That means the cells are big and they have this light pink cytoplasm. Appearance is that of a very light pink kind of an appearance. Okay, so this is how we will see on microscopy, and these will aggregate in the intima, and that will, once you have a lot of these foamy macrophages within the intimal layer, that will produce a yellowish discoloration on gross. That means if you see the vessel wall, you can see that there are these yellow plaques, and that yellow color is because of these lipid vacuoles which is there within the intimal layer. And some of these fat-laden cells may rupture, which will release the lipid into the extracellular space. So once it enters into the extracellular space, these cholesterol esters will crystallize and they will form long needles, which we can see, we call them cholesterol clefts, okay, on the tissue sections. Xanthoma, this is how this will appear. This is how a foamy macrophage is going to appear, this light pinkish color. Xanthoma is another variety of intracellular accumulation of cholesterol within the macrophage, and it is characteristic of acquired and hereditary hyperlipidemic states. And clusters of foamy cells are found in the subepithelial connective tissue of the skin and in the tendons, which will produce tumorous masses known as xanthomas. Next is cholesterolosis. This refers to the focal accumulation of cholesterol-laden macrophages in the lamina propria of the gallbladder. And this is a microscopic finding, basically, in many of these gallbladder which are removed for acute or chronic cholecystitis, when we see the sections under microscopy, we will see that there are these focal accumulations of cholesterol-laden macrophages in the lamina propria. So that particular condition is called as cholesterolosis. Nothing much of significance is associated with this, and the mechanism of formation is also not known. In Niemann-Pick disease type C, lysosomal storage disease, this particular variety is caused by mutations affecting an enzyme involved in the cholesterol trafficking, which will result in cholesterol accumulation in multiple organs. So after lipid deposition, we will move on to protein deposition. Proteins basically in the cell, intracellular accumulation will appear round eosinophilic droplets, vacuoles or aggregates in the cytoplasm. And if we see by electron microscopy, they look like amorphous fibrillar or crystalline in appearance. In some disorders, such as certain forms of amyloidosis, abnormal proteins deposit primarily in extracellular spaces. Intracellular, as I already told you, these rounded eosinophilic droplets, these are basically seen in plasma cell dyscrasias, where we see this round droplet within the plasma cell, and that is called as a Russell body. Okay, so that's basically an immunoglobulin deposit, protein deposit within the cell. Extracellular protein is classical example is amyloidosis. We will learn more in detail about amyloidosis in the coming classes. Then where else can protein accumulate? The accumulation of the cytoskeletal proteins. There are several types of cytoskeletal proteins, which include microtubules, thin actin filaments, thick myosin filaments, and the intermediate filaments. All of these are basically protein accumulations. And accumulations of keratin filaments and neurofilaments are associated with certain types of cell injury. Alcoholic hyaline is an eosinophilic cytoplasmic inclusion in the liver cells that is characteristic of alcoholic liver disease, and it is composed predominantly of keratin intermediate filaments. Like I told you, the alcoholic liver disease passes through multiple stages. The first one was the fatty change, where you have the lipids accumulating there. Next stage is the alcoholic steatosis or alcoholic hepatitis stage, and next would be the cirrhosis stage. Now, in these two stages, in the last two stages, you will see the intermediate keratin filaments will accumulate within the cytoplasm of the liver cells, and that's called as alcoholic hyaline. It appears very, it appears like this pinkish kind of inclusion within the cytoplasm. Then where else can protein accumulate? The neurofibrillary tangle found in the brain in Alzheimer's disease contains neurofilaments and other proteins. Aggregation of abnormal proteins, like these abnormal misfolded proteins may deposit in the tissues and interfere with the normal function. Then the deposits can be, as I already now told you, it can be intracellular, extracellular, or both, and the aggregates may directly or indirectly cause pathologic changes. Certain forms of amyloidosis will fall into this category. Next is the hyaline change. Hyaline change basically is an alteration within the cell or in the extracellular space, which will give this homogeneous glassy pink appearance in the routine histologic section stained with HND. It's not a pathologic change per se. It's a descriptive histologic terms, if I can say. That is, whenever we see certain things, we tend to describe what it is. So one of such descriptive terms is this hyaline change. This is a general term used for anything which appears pink, homogeneous, glassy on our HND sections. Okay, rather than being a specific marker for cell injury, hyaline change is more of a descriptive histologic term. And this morphologic change is produced by a variety of alterations and does not represent a specific pattern of accumulation. Intracellular accumulation of protein, like the Russell body, which I spoke about, which is basically, Russell body is basically seen in the plasma cells. It is immunoglobulin deposits. And alcoholic hyaline, I told you, cytoplasmic inclusions in alcoholic liver disease. These are basically, on a broad umbrella of terms, we can call them as hyaline deposits. And extracellular hyaline has been more difficult to analyze. The collagen fibrous tissue in the old scars will appear hyalinized, but then the biochemical basis of this change is not very clear. So even that is going to be called as hyaline change. The scar, basically after the connective tissue fibers are going to heal. And in long-standing hypertension and diabetes, the walls of the arterioles, especially in the kidney, will become hyalinized, resulting from extravasated plasma protein and deposition of the basement membrane material. Now, this is the picture for that. This is the kidney. Okay, so this is how we can see that there is this deposition of eosinophilic material. This is all hyaline change. Next, glycogen deposits. Glycogen is a readily available energy source stored in the cytoplasm of the healthy cells. Excessive intracellular deposition of glycogen is seen in patients with an abnormality in either glucose or the glycogen metabolism. Whatever the clinical setting, the glycogen masses appear as clear vacuoles within the cytoplasm. And why does it appear clear? Because glycogen will dissolve in our aqueous fixatives. Therefore, basically, whatever tissue we fix in our absolute alcohol, it's going to be dissolved in that, and therefore it will appear like an empty space there. Staining with special stains like best carmine, and the most commonly used stain to identify glycogen is the PAS, that is periodic acid Schiff. This stain will impart a rose to violet color to glycogen. And then glycogen accumulates within the cells in a group of related genetic disorders that are collectively referred to as the glycogen storage diseases. And in these diseases, the enzymatic defects in the synthesis or in the breakdown of glycogen will result in massive accumulation within the cell, causing cell injury and thus cell death. Then the last part of intracellular accumulation is the pigment. Now, once we say pigment, we have two types of pigment, exogenous pigments and the endogenous pigments. Exogenous is all whatever we can think of that can come from outside and which basically cannot be enzymatically degraded by our body because our body is not equipped with such a mechanism, and thus they will be stored as pigments within our macrophages. Okay, that's your exogenous pigment. Endogenous pigments are certain colors which are seen within the cells, and they are produced by the body, our cells. Okay, so see, basically we describe this And some of these terms we will have to know because they appear on the histopathology reports and they will appear as questions in your MCQs. Okay. So exogenous pigments, the most common I already discussed is the carbon pigment, which is there in air everywhere. All our lungs are black. When the child is born, if we do a fetal autopsy or a young kid autopsy, the lungs are seen as very pink in color. But then with age, as much as we inhale our polluted air, the lungs will start accumulating this carbon pigment, and thus it will become black in color. So that's a classical example of exogenous pigment. So when it is inhaled, it is picked up by the macrophages within the alveoli, and then it is transported through the lymphatic channels to the regional lymph nodes in the tracheobronchial region. Accumulation of this pigment will blacken the tissues of the lungs, that is called as anthracosis, and of the involved lymph node. Then where else you will see, apart from our basic pollution, also in coal workers. These coal workers, you know, coal workers lung disease, that's also called pneumoconiosis. They are at extreme risk of developing coal workers pneumoconiosis because of lots of extra amounts of inhaled carbon particles. Okay, carbon dust is inhaled, which will induce a fibroblastic reaction, and then it is so much so that it will induce lung diseases, emphysema, and that is called as coal workers pneumoconiosis. Then of late, I mean, this was there before, nowadays it's become too much of a fashion. How do you think tattooing is going to make sure that that particular whatever tattoo has been tattooed on the skin stays that way? That's because biologically it is a localized exogenous pigmentation of the skin. When the skin is tattooed with whatever picture or whatever writing, whatever pigment is inoculated in the dermal region, no, they are phagocytosed by the dermal macrophages, and this pigment is now not going to be degraded because the dermal macrophages don't have the mechanism or the enzymes to degrade it. Therefore, this pigment is going to sit within those macrophages for the remainder of the life of emblemished. Okay, and the pigments usually don't, that's what is the case in 90% to 95% of the people, that it will not evoke any inflammatory response. Okay, but there are some people for whom tattooing also causes serious consequences, who will hyperreact to this exogenous pigment. There are documented cases, but for all practical purposes, majority of them do not evoke any inflammatory response. That's about exogenous pigment. Then what all endogenous pigments can we think about? One is called a lipofuscin. It's basically an insoluble pigment, also called lipochrome or wear and tear pigment. It is this brown colored pigment which can be seen in almost all the organs as and when we age. So if an elderly patient's sample you will get of any organ, you will see a lot of this lipofuscin pigment. Next is melanin. Melanin, all of us know, is there in all our body. So it is derived from the Greek word meaning black. It's an endogenous brown-black pigment formed when the enzyme tyrosinase catalyzes the oxidation of tyrosine to dihydroxyphenylalanine within the melanocytes, right? So if there is some problem with this enzymatic machinery, you know that the patient becomes albinos. Melanin is needed. It's an endogenous pigment. Then for all practical purposes, melanin is the only endogenous brown-black pigment. The only other that could be considered in this category is homogentisic acid. This basically is not normal. Okay, homogentisic acid accumulation occurs in patients with alkaptonuria, wherein there is a deficiency of this particular enzyme. It's a rare metabolic disorder. In alkaptonuria, this blackish brown pigment is accumulated in skin, connective tissue, and cartilage, and that pigmentation is known as ochronosis. Okay, ochronosis seen in alkaptonuria homogentisic acid. Sorry. Then last pigment, endogenous pigment, is hemosiderin, very, very common. Can be seen in many tissue sections that we receive because it's a hemoglobin derived pigment. It's golden yellow to brown granular pigment, and it's the major storage form of the iron. And hemosiderin deposition, excess of hemosiderin deposition can be seen in organs or in tissues where there has been a bleeding, or in hemolytic anemias where the RBCs are going to have a big turnover and the RBCs are broken down. So whatever is released from that will accumulate as hemosiderin laden macrophages. So that's one thing you have to keep in mind. So that was about the intracellular accumulations and their significance. One more important thing which you will have to remember is pathologic calcification. When we talk about pathologic calcification, there are two types of pathologic calcification which we talk about. One is called the dystrophic calcification, the other one is called as the metastatic calcification. Dystrophic calcification is encountered in areas of necrosis, whether they are of coagulative necrosis, caseous necrosis, or liquefactive, and in foci of enzymatic necrosis of fat. If you remember or recall the class when we spoke about the necrosis, we said that there's going to be calcification in all this. Then in this liquefactive necrosis, especially the pancreatic liquefactive necrosis, enzymatic necrosis of fat, there's going to be calcium which will react with the necrosis and it will form soaps. Calcium soaps are formed in that. So that kind of calcification, that process and that thing is called as dystrophic calcification. Dystrophic means basically calcium which goes and deposits itself on dying and decaying tissues. That is dystrophic. Okay, so if you can see here, this is the leaflet of the heart which has undergone chronic ischemic injury with, I think there is also some left ventricular hypertrophy. Whatever calcification you see here, this shiny, chalky, whitish pigment, what you see here, this is the dystrophic calcification. That is dystrophic, which we will definitely report on our histopath slides. More important for us is the metastatic calcification, because many a times this will pose a diagnostic challenge, because you will not have anything else with some patients presenting with symptoms. Just if you get a calcium done, you will get a very high calcium. So then you will have to figure out why that high calcium is. And in that case, metastatic calcification becomes important. Okay, this is where calcification occurs in normal tissue when there is hypercalcemia in the body. So as opposed to dystrophic, wherein calcium gets accumulated in the dead dying tissues and the blood calcium levels are normal there. There is no hypercalcemia. There is only eukalcemia. That's the difference between dystrophic and metastatic. Whereas in metastatic, there is definitely a hypercalcemia. If you measure the calcium levels in the blood, it is going to be raised and the calcium will deposit in the normal tissues. And hypercalcemia can also accentuate dystrophic calcification. But then, as I said, as a thumb rule in dystrophic calcification, usually the serum calcium levels are normal. Now, why you will have hypercalcemia? So you will have to go back to your biochemistry classes, wherein calcium homeostasis is one big thing which you will have studied about, the axis of calcium. So here I'll just touch wherever you will have hypercalcemia. One, when there is increased secretion of parathyroid hormone. You know that because of increased PTH secretion, there is subsequent bone resorption. And as in hyperparathyroidism due to parathyroid tumors and ectopic secretion of parathyroid related proteins by malignant tumors. So these are the conditions where you will have increased PTH. Second, where you will have hypercalcemia is resorption of bone tissue secondary to primary tumors of the bone marrow, like in multiple myeloma or in leukemia, or diffuse skeletal metastasis like in breast cancer, or accelerated bone turnover like in Paget's disease, or prolonged immobilization. So this can also result in hypercalcemia. Third, vitamin D related disorders, which includes vitamin D intoxication, sarcoidosis, and idiopathic hypercalcemia of infancy. It's called Williams syndrome, which is characterized by abnormal sensitivity to vitamin D. So in such cases, because we know vitamin D is very much essential in the calcium homeostasis, so anything relating to vitamin D disorder will also cause hypercalcemia. The last mechanism is where there is renal failure. So basically, what happens in renal failure is there is retention of phosphate. Now, because the phosphate is retained, there is secondary hyperparathyroidism. And because of that, you will have hypercalcemia. Less common causes include aluminium intoxication, which occurs in patients on chronic renal dialysis, and the milk alkali syndrome, which is due to excessive ingestion of calcium and absorbable antacids such as milk or calcium carbonate. Those are less common causes. So that's about metastatic calcification. Finally, a word on cellular aging. Cellular aging is the result of progressive decline in the cellular function and the viability caused by genetic abnormalities and accumulation of cellular and the molecular damage due to the effects of exposure to exogenous influences. So we know that once a cell is born by mitosis, finally it will undergo aging and finally it will be removed from the body. So that is the course of the usual replicating cell. What are the mechanisms of cellular aging? First, there will be DNA damage. Second, there is something called as cellular senescence. That means the cell also is becoming old. That's a normal phenomenon. Now, under that, we have telomere attrition and activation of the tumor suppressor genes. The third mechanism is the defective protein homeostasis. And fourth is the deregulated nutrient sensing by the cell. Now, what about DNA damage? We know that we have spoken so much about the cell injury that anything exogenous, such as physical damage, stress, chemical damage, biologic damage because of virus, bacteria, etc., they all will damage the DNA. And once the DNA is damaged, it becomes more prone to mutations. And the body's mechanism is such that, it will try to get rid of those cells wherein there has been a DNA damage so that that is not passed on to the next cells. So we have discussed that in detail. So if that is not possible, then the cell will be sent to a killer pathway. It will undergo apoptosis. Next, by endogenous DNA damage by oxygen-free radicals. That is called your reactive oxygen species. That will also result in DNA damage. And what is this term of cellular senescence? So basically, all the normal cells have a limited capacity for replication, and after a fixed number of divisions, the cell will become arrested in a terminally non-dividing state. That is called as replicative senescence. So once a cell is born by mitosis, it will have a particular number of more cell divisions that it is going to undergo, which is kind of fixed. And finally, it will get into something called as a replicative senescence stage, wherein it will be non-dividing. Now, how will the body accomplish this? By one of the methods by which this is accomplished is called as telomere attrition. You all know that whenever a cell is produced by mitosis, there is the small region in the telomere which does not replicate. So with each cell division, this telomere will get shortened and shortened. Okay, so because of the progressive shortening of telomeres, the cell will go into cell cycle arrest. So that's one of the signaling mechanism that once the telomere comes to this particular length, then it will signal a cell cycle arrest. We will learn much in detail about the action of telomeres in the chapter of neoplasia. Second mechanism by which cellular senescence is achieved is by the activation of the tumor suppressor genes. See, basically tumor suppressor genes are like a policeman. They will make sure that nothing which has some mutation will be passed on, so that it is a fertile ground for the tumor to develop. So these policemen will now get activated. There is a lot of molecular biology involved in this, but it is enough to know that there is the CDKN2A locus in the tumor suppressor gene that will get activated, which will activate P16 or INK4A. What will happen once the P16 gets activated? It will signal for replicative senescence. Otherwise, basically, what will P16 do? P16 will protect the cells from uncontrolled mitogenic signals and pushes the cells along the senescence pathway. In neoplasia, you will see that. That means in cancer development, you will see that this P16 gets suppressed. So once P16 gets suppressed, the cell will no longer go into senescence, and it will be like as if it has got a license to keep on replicating. So that's what happens, and thus cancer will develop. But in normal cell, right now we are talking about normal cell and normal cellular senescence, and that's how it's going to happen by activation of tumor suppressor gene and activation of P16. One more mechanism which will contribute to cellular aging is the defective protein homeostasis. Basically, within the cell, the protein homeostasis involves two mechanisms. One is that will maintain the proteins in the correctly folded conformations. You know that whenever there is a misfolding, it will cause autophagy, right? That's what happens, where those which will degrade this misfolded proteins by autophagy lysosome system. Now with aging, what will happen is that both these pathways will be impaired. Abnormal protein homeostasis can have effects on cell survival as well as replication. And once there is this starting off, accumulation of misfolded protein will trigger apoptotic pathways as well. So basically, protein homeostasis, if it is tipped in either way, will result in cellular aging and destruction of that particular cell. Last is the deregulated nutrient sensing within the cell. In the cell, there are two major neurohormonal circuits which will regulate the metabolism. One is the insulin and the insulin-like growth factor 1 signaling pathway. Insulin-like growth factor 1 is produced in response to growth hormone by the pituitary, and it will produce and it will promote anabolic state, cell growth and replication. Okay, so if this insulin-like growth factor 1 is reduced, that means the cell will move towards aging. There is something else called as sirtuins. Sirtuins are a family of NAD-dependent protein deacetylases. These are thought to promote the expression of several genes whose products will increase the longevity. Which are these proteins? These include the proteins that will inhibit the metabolic activity, reduce the apoptosis, stimulate the protein folding, and inhibit the harmful effects of oxygen-free radicals. So all these anti-aging creams and all that, when they say the cell is going to be there for a longer period of time, increasing the longevity, when we talk biochemically and molecular pathology wise, this is what it is supposed to do. It is supposed to, increase the release of certain sirtuins, which will promote the expression of several genes, which will result in, this good kind of environment within the cell. With this, what will happen is if there is caloric restriction, if the person is consuming less calories, that will increase the longevity by both reducing the signaling intensity of interleukin 1, reducing the signal intensity of insulin-like growth factor 1 pathway, and by increasing the sirtuins. Now, sirtuins is an area of research now, wherein they are looking at trying to replicate and increase sirtuins so that the longevity increases. It's still an upcoming and ongoing research as can be expected. So this is the chart of the mechanism that will cause and counteract the cellular aging. So we said first that there will be DNA damage. DNA damage could be because of physical agents, chemical agents, or biologic agents, carcinogen exposure, or maybe endogenous reactive oxygen species. If there is a DNA damage and the defective DNA is repaired, then the cell will be allowed to replicate. But if the DNA is repaired, then the cell will be allowed to replicate. But if the DNA is not repaired, there is a defect in the DNA repair genes, then that particular cell, because of this DNA damage, will become mutated. And because it undergoes mutated, it will be sent to a killing pathway. That is, it will become cellular aging. Finally, it will die. Apart from DNA damage, we have cellular senescence. How will cellular senescence be done? One is by telomere shortening, which will reduce the cellular replication, cell loss, and again it goes into cellular aging. Then defective protein synthesis, protein homeostasis. I already explained to you how the protein homeostasis about the correctly folded protein, and if at all there is a misfolded protein, it will be sent for cellular aging and the cell death. And then we have nutrient sensing. Nutrient sensing will basically reduce the insulin-like growth factor 1 signaling, and it will increase the sirtuins. Then there will be altered transcription, which will result in increased DNA repair and increased homeostasis. But if this is not done, then it will go in for cellular aging. That's about cellular aging. Now we will come to MCQs. If any students are there and they have any questions, you can ask me. Do we have students? Yeah, if nothing is there, then I will continue with the MCQs. A 71-year-old male diagnosed with pancreatic cancer is noted to have decreasing body mass index. His normal connective tissues undergo atrophy by sequestering organelles and cytosol in a vacuole, which will then fuse with the lysosome. However, the cancer continues to increase in size. So which of the following processes is most likely occurring in the normal cells but is inhibited in the cancer cells of this man? So what is happening here? They say that his normal connective tissue is undergoing atrophy by sequestering organelles and cytosol in a vacuole and fuses. So this is your basic autophagy and phagolysosome theory. That's happening within the normal connective tissue. But the cancer tissue is continuing to grow, right? So what are they asking you? They're asking which is most likely occurring in the normal cells and it is inhibited in the cancer cells. So the options are aging, apoptosis, autophagy, hyaline change, or karyorrhexis. Any answers?
Autophagy, ma'am.
Autophagy is occurring, correct. But which particular process is occurring in the normal cell and inhibited in the cancer cells? Whatever in the context of what we have learned today.
Aging.
Basically, aging.
So I am aging.
Yes.
I am
Yes.
aging.
Yes, it is aging. Okay. Basically, the normal cell is still having aging and cancer cell has evaded the process of aging. It has evaded aging. It has evaded. See, in aging, everything comes. Apoptosis, autophagy, everything. So cancer cells have very cleverly evaded all this and collectively they have evaded aging. So the answer to the question will be aging. Okay. Next question. A 69-year-old woman examination, she has an audible bruit on auscultation of the neck. Is performed. The curated atheromatous plaque has a grossly yellow tan, firm appearance. Scopically, which of the following material can be found in abundance in the form of crystals within the cleft-like spaces? Am I audible? Is my video or audio glitching? Can you please let me know?
Yes, no problem. No problem.
No problem. I'll continue. Okay. I just got a message about internet. That's why I thought I'll ask you. They are talking here about an atheromatous plaque. I think by now you know that there is an atheromatous plaque, and they're asking you, when I see that plaque under, and they've told you grossly it is yellow tan. Okay. When I take a section of that and see it under the microscope, what can be found in the form of crystals and cleft-like spaces? Cholesterol, glycogen, hemosiderin, immunoglobulin, or lipofuscin?
Cholesterol.
Correct. Correct. Hello, am I audible?
Yes, ma'am.
Cholesterol. The answer.
Audio is not clear, ma'am.
Cholesterol clefts. I explained it to you that, cholesterol cleft, thin, long, needle-shaped crystals, cleft-like spaces. So these are called cholesterol clefts. Voice not clear?
Yes, ma'am. Audio is getting interrupted.
Okay, let me see. 1 second, just give me a second. Now, how is it now? It is better?
Yes, ma'am.
Yes, ma'am.
We just have some more questions to go. At autopsy, the heart of a 63-year-old man weighs only 250 grams. What is the normal? It is 330 grams. And it has a small right and left ventricle. The myocardium is firm with dark chocolate brown color throughout. The coronary arteries show minimal atherosclerotic changes. An excessive amount of which of the following substances shown in the figure would most likely be found in the fibers of his heart? Is it bilirubin, glycogen, hemosiderin, lipofuscin, or melanin? Sorry, the picture I didn't get, but the question is quite straightforward. They're talking to you about quite an elderly person's heart, and they say that heart has undergone atrophy. And they very clearly say that the coronary arteries show minimal atherosclerotic changes. But also
Lipofuscin.
Sorry?
Lipofuscin.
Lipofuscin, correct. Dark chocolate brown color throughout is basically the wear and tear pigment they are talking about, and that is lipofuscin. Bilirubin easily ruled out, glycogen nowhere near, melanin not doesn't come into picture. You may have confusion between these two, but then hemosiderin you can expect whenever there has been extravasation of RBCs and some bleeding. This is a normal heart which is atrophied. So it is a wear and tear pigment which we are talking about, and it is lipofuscin. 69-year-old woman has had a chronic cough for the past year. A chest X-ray reveals a 6 centimeter mass in the left lung. A biopsy specimen of the mass shows carcinoma. A pneumonectomy is performed, and the examination of the hilar nodes reveal uniform surface. Which of the following factors most likely account for the appearance of these lymph nodes? See, basically they're giving you too much sentences is actually too much of information. You can see with the options: aging effect, bleeding disorder, cigarette smoking, liver failure, multiple metastasis. See, they're trying to confuse you by saying that he has a lung cancer. Basically, this whole sentence means this is a 69-year-old man. The age is a little important. He's developed a malignancy in the lung. Okay, that's what is the gist of these two sentences. Then they have sent you lymph nodes. Okay, you're not talking about the lung. They're talking about the hilar lymph nodes. And what was it showing? It's showing a uniform dark black cut surface, right? So what could be the reason of that dark black cut surface? Is it aging, bleeding disorder, cigarette, liver failure, or multiple mets? Do you think it is metastasis? Will metastasis have color? We haven't done neoplasia, I agree. But metastasis will not have color. Okay.
No, ma'am.
Okay, it will be, if at all you see, you can see some necrosis. Okay, but then you will not have black color. So what is the next most appropriate answer in this? No? Still confusion? Confusion between which options?
Ma'am, A and B.
See, bleeding disorder, if we're talking, we will talk about hemosiderin-laden macrophages. Hemosiderin-laden macrophages will have a distinctive golden yellow to brown color. Okay,
Yes, ma'am.
not a dark black. What else was black when we spoke about? Aging effect would be lipofuscin, right? Wear and tear pigment, brown. What else was black when we spoke about? I didn't give this example, but then common sense. What will make our lungs black?
Ma'am, cigarette.
Yes. Air pollution, carbon particles will make the lungs black. Once the lung is black, basically the macrophages are going to drain into the lymphatics only, no? So once they drain into the lymphatics, they will end up in the hilar lymph nodes only, right?
Ma'am, it's given 6 centimeter is mass 1.
That is different. That's what I'm saying. They have confused you. Deliberately, the question is confusing. The first 3 sentences just say that he has a mass in the lung. Why is it? This is also connected. Why is he having mass in the lung? Why is he having a cancer of the lung? Because he is a chronic cigarette smoker.
Yes, ma'am.
Understood?
Yes,
I
ma'am.
hope this is clear, you know, and they're talking about lymph nodes here. They are telling you that the lymph node, hilar lymph node, uniform dark black cut surface. And they're asking you, what is this which is responsible for this black colored appearance in the lymph node?
Yes, ma'am.
Right? So basically it is cigarette smoking. And that indirectly can be considered as a cause for his cancer lung. Yeah? Clear?
Yes, ma'am.
Clear on this? The question, see, the answer is very straightforward. If you were given a one-liner question saying that the black color of the hilar lymph nodes is because of, all of you will answer, correct? This is a clinically based question so that it will confuse you. The answer they want is actually straightforward, but they've given you extra information so that they are misleading you. Okay, please read the questions carefully. That's my biggest suggestion. Read the questions carefully and take the relevant points given in the question for you to answer that. Okay. So the answer here is cigarette smoking. Basically, it is the dark carbon pigment. That's all it is. Okay. See, liver failure will not give you any color. Bleeding disorder, I explained, it will be hemosiderin-laden macrophages, golden yellow to brown. Aging effect will be, if at all I can extrapolate it, I can say wear and tear pigment, and that is lipofuscin. We know it will be brown in color. Metastasis as such will have no color. Unless there is some necrosis, necrosis will be yellowish in color. Okay. Yeah, one more question. A 22-year-old woman from Albania has a congenital anemia requiring multiple transfusion of RBCs for many years. Okay, at 22 years, she has undergone multiple transfusions. Keep that in mind. On physical examination, her skin has a bronze color. Liver function tests show reduced serum albumin. Which of the following findings would most likely appear in a liver biopsy specimen? Amyloid, bilirubin, glycogen in the hepatocytes, hemosiderin in the hepatocytes, steatosis.
Hemosiderin.
Option D.
Correct. So you have now understood, right? Whatever information they have given, what to take in that. Basically, they're trying to tell you that she's had some hemoglobin.
That is this.
Yes, yes. She's had some hemoglobinopathy for which she's been getting multiple transfusions. And we know that people with multiple transfusions will end up with
Iron overload.
acquired hemosiderosis. Okay. So hemosiderin pigment is there in her hepatocytes. Okay. What color will amyloid be? Amyloid will be pink. Okay.
Yes.
Pink amorphous acellular material, amyloid. Bilirubin will be yellow. Glycogen, how will glycogen look under H&E sections? Empty space, because glycogen dissolves in our routine absolute alcohol sectioning. So to demonstrate glycogen, we will use periodic acid shift, that is PAS stain, and there it will be red violet in color. Okay. And what is steatosis? Steatosis will appear like, it's basically the fatty change. You'll have these empty spaces. Okay. And the special stain to demonstrate fat is called oil red O. Okay. Like how we have for glycogen, we have PAS. To demonstrate fat, it is oil red O and Sudan black. These are the two special stains used to demonstrate fat in sections. Yeah. Now this is one more question. 72-year-old man died suddenly from congestive heart disease. At autopsy, his heart weighed about 580 grams. Normal is 330. That means you can easily say that there's been some hypertrophy, right? And it showed marked left ventricular hypertrophy and minimal coronary arterial atherosclerosis. A serum chemistry panel ordered before death showed no abnormalities. Which of the following pathologic processes best accounts for the appearance of the aortic valves seen in the figure? See, he has had congestive heart failure, and because of which he has died. So you have left ventricular hypertrophy and you have this kind of appearance of his aortic valve. And they say it is not atherosclerosis because very clearly they said minimal coronary arterial atherosclerosis. And serum chemistry panel before death is normal. Okay, so what could this be? Is it amyloidosis? Is it dystrophic calcification? Is it hemosiderosis? Is it hyaline change, or is it lipofuscin deposit? I think there's one more option. Sorry, I gave away the answer. It is dystrophic calcification. Yes. Okay, so it's straightforward. By now you have understood, right? This is a very straightforward case. You can see nicely that the calcium is deposited on his aortic valve, which has undergone some necrosis because of congestive heart disease. And they say that the serum chemistry before the death is normal. That means his calcium levels also, we have to assume that it was normal. Okay. Next. A 70-year-old man with hypercalcemia died suddenly. So first clue, there is hypercalcemia. Okay. At autopsy, microscopic examination showed non-crystalline amorphous deposits of calcium salts in gastric mucosa, renal interstitium, and alveolar walls of the lung. So multiple organs are involved. Which of the following underlying condition would likely explain these findings? Is it chronic active hepatitis? Is it diffuse parathyroid hyperplasia? Disseminated tuberculosis? Generalized atherosclerosis? Or normal aging process? So the biggest clue in this is that there is hypercalcemia.
Metastatic calcification.
Correct. It's a question on metastatic calcification. Very good. So they have also told you that there has been deposits of calcium salts in multiple organs. So where can we have hypercalcemia leading to deposits in multiple organs? Something to do with the calcium homeostasis, right?
Hyperparathyroidism.
Hyperparathyroidism. Very good. So the answer is
Diffuse.
Sorry, there was one more option, but then irrelevant one. Diffuse parathyroid hyperplasia. Okay, we'll have to assume that hyperplastic parathyroid increased release of parathyroid hormone, which has resulted in hypercalcemia, and the effect of parathyroid hormone with hypercalcemic deposits in all these end organs, that is gastric mucosa, renal interstitium, and the lungs. Okay. An experiment analyzes cells for enzyme activity associated with sustained cellular proliferation. Which of the following cells is most likely to have the highest telomerase activity? They're asking you which of the following cells will have the highest telomerase activity. Is it the endothelial cell? Is it the erythrocyte? Is it the germ cell, neuron, neutrophil? So what is telomerase activity? Telomerase is the enzyme which is responsible for the shortening of the telomere.
Aging process.
In the aging process. So when the cell is new, then it will have a very good amount of telomere. And then as and when the cell goes into aging, the telomere gets shortened. That means your telomerase activity will also be seen in that. Yeah. So now you tell me which of these cells will have highest telomerase activity. That means which of these cells have highest propensity to undergo division. That means highest mitosis. One option immediately you will rule out, which is the end cell in this, which will not divide, which is the non-dividing cell. Neuron. Yeah, you rule out neuron, okay? Because it's a non-dividing cell. Next, there is one more cell which has no nucleus. So its lifespan is fixed. It is not going to replicate. Which is that?
Erythrocytes.
Erythrocyte. Yes, very good. So you have now ruled out erythrocytes and neurons. Neutrophils. How about neutrophils? What do you think, neutrophils? Do you think they will keep on replicating?
No, ma'am.
No. How do the neutrophils come? They come from the bone marrow. You have the myeloid series in the bone marrow, which will produce from the myeloblast. It will become promyelocyte, metamyelocyte, myelocyte, band form, neutrophil, and then finally it will help in maintaining the police keeping of the body, and finally it will die. So you have ruled out your neutrophil also. Now what is left? Left is the germ cell and the endothelial cells. Correct?
Yes, ma'am.
Now apply your common sense. They are asking you highest telomerase activity. That means which will have highest propensity to divide, more and more divide. Which cell?
Germ cells.
Germ cells. Correct? Agree?
Yes,
Sounds
ma'am.
good? Sounds good. Endothelial cells also will not divide. Okay? So the answer is germ cell. So here we arrived by exclusion, because telomerase activity and all that, molecular talking will sometimes mislead you. The questions where you find that there is too much of molecular pathology coming in, you need to associate which enzyme, where it is going. So many times you will get blinded by that. So there is another way in which you can still come out with a correct answer by exclusion. Okay, you'll have to keep on excluding the options. Finally, at least you need to come down to 2 options. Then it will be easy for you to read the question again, understand it, and opt the correct answer. Yeah, that's the germ cell. Any question on this? Understood the question? See, experiment basically wants to analyze enzyme activity associated with sustained cellular proliferation. Germ cells, we know, are the ones which will keep on dividing, right? That's your pluripotent stem cell. It can divide and differentiate into anything. So that is the one which will have your highest telomerase activity. Yeah. One more. A study of the aging shows that senescent cells have accumulated damage from toxic byproducts of metabolism. There is increased intracellular lipofuscin deposition. Prolonged ingestion of which of the following substances is most likely to counteract this aging mechanism? So clearly they have told you there is aging going on. There is an increased intracellular lipofuscin. Now they are telling you prolonged ingestion of which of these following is most likely to counteract this aging mechanism. So they're asking you something which is going to counteract aging. Antioxidants, analgesics, antimicrobials, antineoplastic, glucocorticoids.
Antioxidants.
Antioxidants.
Everybody knows, even advertisements tell you this, no? Correct. You don't have to study pathology to know this. Even advertisements, every skin cream and every, I think, shampoo will tell you antioxidant everywhere is hanging around. Correct? So the answer is antioxidant, but then ingesting antioxidants is that tough. Basically, you'll have to eat a lot of vegetables, fresh vegetables and fruits. Yeah, that's it for today's class. If you have any questions, you can ask me or you can put it up in the chat box. Anything? Wanna ask me anything?
No, ma'am.
Yeah. Okay, then we will conclude today's class. Next class, I will start with a new chapter. Okay.
Thank you, ma'am.
Yeah.
Thank you.
Thank you. Bye.
Thank you, madam. Thank you all.
Thank you.